So, hi everyone. So I will try to present the work undertaken for several years for conservation of a rare palm endemic to New Caledonia. So my apologies for my English. I learn on Netflix, so it will be bad sometimes. And I have to read my notes, sorry for that. So I'm going to give you some results of this study still in progress, and for which I have a reason for work for 2 years. I represent the New Caledonia Agreement Institute that works in addition to agricultural and human science on restoration of mine sites with common and endangered species. We have a specialized laboratory on seed ecology. So here we go. So the beginning, welcome in New Caledonia. So we have white sand, nice blue water, and red soils. So red soils are ultramorphic soils. Actually, more than a third of the Grande Terre, the big island, this one, are covered with ultramorphic soil. And sorry. And the south, mainly the red soils are in the south here, and all along the mountain chain. So ultramorphic soils are rich in toxic elements for plants and poor in essential elements. So this particularity of the soil reflected by adaptation of plants in the soils, which in some places more than 19% of endemism. Cerebus gianei is one of these plants, a palm. Also, he is only indigenous representative of the Livistonia genus and subtribe in New Caledonia. So we are in the south, and very sorry. Only on so the Cerebus gianei grow only on ultramorphic soils in the south of New Caledonia, and it was subject to an intensive harvest for its edible art by prisoners of penitentiaries in 1890s. So disappeared since the end of the 19th century. So in 1980 we found it. Yeah, great. So thanks to the hunter. So in the south. So after discovering the Cerebus, rediscovered the Cerebus, the Cerebus was not monitored and no conservation program was taken, and as a result of the first fructification around 1982, seeds were sent to different botanical gardens. So between 1980 and 2009, sorry. So there was a lot of exchange, but we don't have a lot of data. So we know that there are some plants in Hawaii, Florida, France, Thailand, and Queensland. So yeah, there is in Queensland. So I'm watching you all. So all this traffic is very confidential, and sources are never really reliable. It's difficult to estimate the number of cultivated Cerebus in the world. So in the world, maybe there are 10 or more. In New Caledonia, 10 plus, and in wild, just 1 mature and 15 juveniles. So that's why in 2009, that work was undertaken with partners. So how we proceed? So to work with the seed ecology and germination, that's my part, and the institute. So for technical and financial support, we have the Valley Company. It's a mine company in New Caledonia in the south. And for the ex-situ conservation area, we have a technical support of le Parc de la Province Bleue. So this site, it's we choose this site because of the condition. The conditions are similar to those observed in the original stand area. So all together, we are strong. So after that, that's harvesting time. So that was not easy as for the plants we usually walk. Because to reach the seed plant, you have to walk in the dense wet forest. And here everything is fine, but on the slope over 60% of inclination and 3,000 meters of altitude. In addition, the only wild palm is about 8 meters long, so here's a little drawing, so no judgment, representing the technique currently used. So you can see a man climbing here and me walking here on the rock. So the technique currently used with cinnamon climbing to protect the precious seeds, and currently a cable is stretched over the palm, so we can attach and reach the fruit without the risk of damaging. So previously a net was put below, no, sorry, just here, but that was not very cool. So you can see what's it in reality. So we did that to have the seedlings in the basket of resistance beginners closer to the natural condition of maturity of the fruits. So it's for the protection and maturity. So the harvest data since 2009, there have been 5 harvests. So 11, 15, 50, 51, 32, and 45. So it's too early to say if the protections allows this increase of fruits of its random, of its cycle, natural cycle, or something else. So as in previously, the technique as we used evolved in a better way, because the first time we lost 8 seeds, not me, but the team before. After that, in 2011, we saw some bite marks, and after, since 2030, sorry, we use oyster mesh bags, and that's perfect for this for conditions. So when we harvest, after we have to measure weight and focus all of the fruits and the seeds. And we have not a perfect cycle, but close to the perfect cycles. You can see on the table. So there is a big difference in weight fruits. Can explain with the maturity since 2013. So after that we can describe better than ever all of the parts of the fruits and seeds. And we saw a positive so it's positive correlated between the fruit size and the depulp size without the mesocarp, and the volume and the height and the weight of the fruits. So we can see we have a big range of weight in the sides in the fruits since 213 and 13, yeah. So we can see all of the measures since the beginning. So that's pretty stable, maybe between the length, weight, and height, but not in weight. Sorry. So after that we put in germination germination test. Sorry. So 3 conditions were used in controlled conditions in between 20 and 30 degrees, greenhouse conditions, and in situ, and just close to the palm tree. So here we are. We have the data for all of the year. So you can see it's very. You have a big difference between between year per year. Sorry. So germination outcomes. Now it's possible to characterize more finally seed germination of Cereus generi and the various associated phases. Sorry. And the first stage of germination is associated with the development of the embryo and the seed until the appearance of the cotyledonary pistule. Sorry. So you can see the cotyledonary pistule blow up the operculum here. So in the picture here, we can see all of the parts of the emergence of the seedling. So in Cerebus generally, it's a remote tubular. It's a distant germination because of the long epiphyll and cotyledonary sheath. So sorry. So all of the family, Arecaceae family, are hypogea. So the seed were not raised above the soil, as you can see on the picture. So for the germination test, to begin with the result obtained without acid gibberellic acid in 2009. Sorry. So for germination with and without gibberellic acid, we don't see a significant difference. So that's why after we put some 3 conditions in a germination, so in situ control and half control. So for the result for the control, the best result, sorry, we had, that was 100% in 217. So in situ, we have just, since the start of the study in 2017, only 1 seed of the 10 seeds we put in the germination. So what? Okay, so I'm gonna be, okay, so you can see all the difference. We have an increase of days before germination by year. So in 2013 we are at 70 days in 20 and 30 degrees, and now we are 107 days. So I have to move fast. Okay, cool. So we saw a big problem in cultivation since the 210, June 210. So we have a slow growth. So as you can see on the picture. And since the beginning, we have a problem with a disease. Maybe it's a Petailopsis. We have to investigate in this. So now the best result we have is when you're using baking soda, and we stop irrigation by propagation. So what we learn? Here we go. So ecologics of Cerebus generally, palatability for fruits, bats, and rats. Good percentage of germination in controlled and greenhouse condition. We need more days to germinate since 211. High mortality percentage after germinations, it's above 70%. I didn't so. High accessibility to disease in nursery and slow growth since 2011. So what we next search of the plants in wild, need to protect and survey the adult tree, focus on germination test. We have to focus on germination test only in controlled condition, constant and maybe hot temperatures. Need to investigate with why we have a high mortality percentage. And to high sensibility to disease in nursery, we need no sprinkler, draining substrate, heat, and we will remove and burn sick leaves. And we need to investigate why we have slow growth since 2011. So thank you. Sorry.
That was perfectly timed to walk. Certainly uncovered a whole bunch of interesting questions.
It's okay. No, it's okay.
Next, we've got Jenny O'Mara talking about endangered species in Brickpit at Sydney Olympic Park. I'm curious now.
Just go left and right. Okay, thank you. Thanks everybody. It's really exciting to be here at this conference today. It's my job to take you through one of the restoration projects we're undertaking at Sydney Olympic Park. It's a long-term restoration project and also involves some long-term monitoring to assess the success or non-success of that restoration project. So what we're trying to do is manage the habitat of endangered species in the Brickpit, and of course our challenge is how to remove invasive environmental weeds out of that habitat when the threatened species really love it. So we've got a diverse range of fauna, including the endangered green and golden bullfrog in the Brickpit, and this is the story about how we're attempting to manage that space for them. So Sydney Olympic Park is about 435 hectares of land, about 14 kilometres west of Sydney, surrounded by a sea of urbanity. And the Brickpit is a 27-hectare property within that space. So as the name suggests, it was a brickpit once, and from 1911 to 1988 it was an operational brickpit extracting shale and sandstone for building operations in Sydney. When the brickpit finished and operations closed down in 1988, that's when they got the idea to run the Sydney Olympics in that area, and there was a lot of work involved in the setting up of venues and rehabilitation of much of the area to create the parklands of Sydney Olympic Park. And the Brickpit was identified very early as having very high biodiversity values. So by the time the Olympics came in 2000, the Brickpit still looked remarkably like a brickpit. It had colonising vegetation, but there was no management. There was a lot of monitoring of the green and golden bullfrog that was present on the floor, but basically an industrial landscape, and our favourite started to arrive. You can see that it's just scree slopes, skeletal soils, and you can probably see a heck of a lot of pampas grass all over the floor. So the ecological values, the green and golden bullfrog was officially discovered on site in 1993. It was right across the Olympic site, but the Brickpit was seen as the core habitat of the frog. So it was a protectable, contained space within which the frog was readily breeding. There was a large proportion of the population down there. They could put a fence around it and protect it. So the Brickpit remained the Brickpit right throughout the Olympics. We've also got quite a large proportion of woodland birds residually living within the Brickpit area as well. So it's valued for its woodland birds as well. Now also having large open spaces, large wetlands, it also attracted migratory species, so Latham snipe is in there. And also lots of vagrants pop in and out depending on regional and statewide weather patterns. So we get lots of very interesting raptors, bitterns and all sorts of things popping up in there. So it is valuable and relatively undisturbed because we still don't let people in there. It is a safety risk. Collapsing walls, deep drowning issues, things like that. So the culprits that moved in, apart from the bullfrog, were our typical subjects. So lots of lantana colonising the walls of the Brickpit, profusion of pampas across the scree slopes, the rubble and other very revolting environment down there, and Juncus acutus. And of course, each one of these is quite important to the biodiversity down there. So lantanas, really awesome for woodland birds. The pampas grass provided excellent sheltering habitat for the bullfrog, particularly during winter. Nice and moist in there, and you can get jolly large ones down there. And in the wetlands, Juncus acutus was very important sheltering habitat in that space. So very important to the frog in that space. So when Sydney Olympic Park took over management, we actually started a vegetation management program. the first thing that was initiated was studies into how the frog and the bird were interacting with the vegetation. So universities, PhD students brought in, consultants did small vegetation studies, so sampling of pampas grass and were the frogs in there, weren't they in there, to what proportion were they in there, and control, so trying to limit those species to the spaces that they inhabited at that point. So removal of all juveniles, so the weed sweeps on a regular basis, taking out all the babies, and removal of seed heads too, which was quite amazing. Visualise 2,500 kilograms of pampas seed heads removed every year. So, and Juncus acutus. I don't know if anybody would like to volunteer to remove Juncus acutus seed heads once a year. We went through lots and lots of companies each year. They wouldn't come back. In 2006, though, after this period of establishment and knowledge gathering, we instigated a long-term staged weed removal. So we started to move into an active phase. We knew where the frog was. It's pretty obvious where the birds were. And so a stage program was initiated on each one of those species. There were other weeds in the Brickpit, of course. There was about 30 species that under law we were required to remove, and those were dealt in different ways. None of them contributed very much to the habitat of our biodiversity as much as these three species did. So one thing we had to do, though, was ensure that we had no net loss, particularly for the bell frog. So before we started removing habitat for them, particularly in the wetlands, we had to have provided more habitat. So the years before 2006, there was planting of Balmia, Chenoplectus, species like that, to provide habitat when we took away Juncus. We were experimenting with plants on the skeletal soils and which plants survive, which has the growth form that's similar to pampas grass. Other things that really slowed things down were the fact that we couldn't use pesticides due to the presence of the frog. No glyphosate could be sprayed willy-nilly in amongst all these fabulous weeds. We could cut and paste. And to make it even more complicated, if we were going to remove frog habitat, we had to do it in a staged manner as well. So a little plot of grasslands or wetlands, you have to cut things down to 300 mil, remove the thatch, come back the next day in a disturbance regime, cut it down to 150 mil, take away the cut thatch, come back the next day with ecologists, make them crawl through it and check for any remaining fauna, and then you can take it down to the ground and remove your plants. So quite intense and quite costly, but it did introduce habitat mosaic for the frog. So the green and golden bell frog is a generalist. We don't actually know what it wants in particular. It tends to move around the landscape a fair bit depending on seasonality and weather conditions. So we try and provide it with quite a range of different aged habitats, open water, closed water, emergent, banks, all sorts of things that we can think of that the frog may like. So the disturbance regime also was seen as important. So the frog seems to like ephemerality, so it will move out of permanent ponds under suitable conditions and take advantage of new ponds. So if that doesn't happen, we actually come in every 3 to 5 years and we rejig our ponds. We take out all the vegetation and reset them ourselves, just giving them that mosaic of maturity. And then there's hygiene protocols. The Brickpit doesn't have mosquito fish, which is a threatening process on the green golden bell frog, predatory. And of course control of the chytrid fungus, which can kill the frog in great numbers. So what we were doing was, apart from this action, we also had to make sure that the new plants didn't provide habitat, so everything had to be removed before it reached a certain size. So all woody weeds couldn't reach a square metre, baby pampas couldn't get more than 10 centimetres, Juncus acutus all had to be removed before 15 centimetres in height, or their base was 5 centimetres across. And all of this had to comply with our protocols underneath the at that point, the National Parks legislation. It's now under the Biodiversity Conservation Act. That was fun. And also we divided the Brickpit up into disturbance units. So you could have a certain amount of disturbance in Unit 14, but you couldn't do the same amount or an equal amount in the next door unit, number 12. So we were trying to spread our disturbance evenly across the Brickpit and not hit any one space too hard. This is what we look at today. This is us in the environment team drawing lines on a map. We manage quite intensively the habitat down there, and on the floor of the Brickpit, so I dare not use this button because it'll be, where's the thingy? Oh, no. Yes. Anyway, on the floor of the Brickpit we have a grassland. So we manage that for grassy habitat only. We remove trees. And on the slopes we have woodland for the birds. We only plant shrubs and ground covers in the Brickpit. Shrubs and ground covers for woodland birds and grassy habitat for the frog. So monitoring is key to all of this, the feedback that whether or not we're being successful. And for the green and golden bellfrog, it's been monitored since 1996. And we have an annual monitoring program which measures abundance, breeding indicators, and where the frog is, what ponds, what terrestrial habitat is it occupying. At the same time, we've encouraged other research to help us try and understand habitat usage and what the animal needs. Hasn't been successful yet. Woodland birds, we have a vegetation assessment every 4 years, quadrats which we measure vegetation density and whether or not the birds are inhabiting it, and an annual spring count which gives us a photograph across the site of where our animals are. This is an example of the frog monitoring. We microchip our frogs so we know exactly where they are and exactly how old they are and whether they're moving from place to place. So pretty intensive, but monitoring has shown that the bullfrog really enjoys the larger permanent ponds and will then move out of that when we have ephemerality, taking advantage of fresh ponds, probably with surges of algae and productivity for the tadpoles. The males are using the planted vegetation in the wetlands, replacing the typha. So a lot of those dots you saw in the previous slide are where juncus used to be, and we've replaced it with native species. The frog also prefers closely connected ponds, nothing greater than 50 metres, thank you. Now, we have achieved no net loss, and we don't use anything more exciting than Juncus kraussei, Lomandra longifolia and Garnia, because they are tough species. It's revolting on the floor of the Brickpit for a plant. We also haven't finished removing all of these weeds. We wanted to finish in 2018, but the growth of the terrestrial plants on the floor has been too slow for us to keep going in the pattern that we wanted to. So we are running probably 50% under for pampas, and juncus has been removed entirely, which is fabulous, and we're removing our last lantana hopefully next year. It was to be this year, but the vegetation and with the drought in New South Wales hasn't been terribly profitable. Have I run out? Woodland birds. As the shrubs get denser, you get more woodland birds. So we've seen that woodland birds is closely related to high shrub cover, low tree density, and wherever there's no noisy miners. Of course, we don't have noisy miners in the Brickpit because there's no eucalypts, and we're going to keep it that way. So if we had a shrub planting program, the birds came, and we were surprised at how quickly the bird habitat was being utilised by the birds. Finches were quite keen and would jump into hakea that were about a year old as long as it could support a nest. So thank you. A lot of the data that's been gathered over the years by many people have been really important to interpret how successful we've been and to guide our program over the years. All of the Sydney Olympic Park staff, Mike Marnie and his team from the University of Newcastle put in a lot of time. Tony Saunders and his Woodland Bird Project, Peter Davies and David Nipperas, who are excellent at statistics at Macquarie Uni. So thank you very much.
I'm sure there's a whole bunch of interesting
Before I start, I'd also like to acknowledge the traditional owners, prior, present and future leaders on the sites that we meet here today. My talk is really on maximising ecological restoration through a threatened species program. This site here at Numinbah has been referenced a few times today through the plenary talks, and I want to demonstrate a little bit more of some of the fabulous results that are occurring out there due to a threatened species program. So the background of the project occurred through the raising of Hinds Dam on the Gold Coast, which impacted about 500 hectares of native vegetation, including 313 hectares of koala and remnant vegetation. In that process, it also impacted and inundated excuse me, 6 nationally environmental significant species. So the onion cedar, spiny gardenia, the 2 macadamias that occur on the southern part of the Gold Coast, the shiny plectranthus and the shrubby jasmine. So at that time, when I did work with City of the Gold Coast, I made it a point to jump on the technical advisory group for the project and directly influence and hopefully be involved in that program. And essentially that facilitated the development and design of the Threatened Species Program in conjunction with other experts, negotiation of the sites, so those receiving sites that they would go on to, and there was actually 2 receiving sites. I'm going to concentrate on one today. The development of a 10-year budget, which is, I think all of us know in this room, is crucial to seeing some success at least, and sourcing additional and strategic land for the purchase of an offset, which actually occurred by linking Springbrook and Lamington, so really key to this program. So we developed a whole lot of plans, including a restoration plan, updating the management plan, and some strategic fire planning, as well as in situ fire planning as well. So a little bit of background on Numinbah Conservation Area. It's a 590 hectare site owned and managed by City of the Gold Coast, purchased in 2002 with the Conservation Levy, contains 12 regional ecosystems and 3 of those are listed of concern, and it connects Springbrook with the Numinbah Valley via also including another valley which Waterfall Creek runs through. 465 flora species identified so far, 28 fauna species, of which 28 that are considered either endangered, vulnerable or rare. And that vegetation on site ranges from rainforest, sclerophyll systems all the way through to some open paddock. It's pretty degraded to start with due to previous logging disturbance, cattle grazing and then subsequent weed invasion. So works began there in 2008. Part of the funding for this project really assisted getting a really good bushfire management plan underway, and that was essential to protecting these threatened species, not only the ones that were being propagated and installed on site, but also to protect those sensitive areas that they were being placed into, as well as those buffering areas around them. Obviously it helps facilitate the protection of life and property as well. And then a restoration plan was developed and adopted in 2010, which contains all the strategy for the whole site. So how to restore the whole nearly 600 hectares as well as manage those threatened species. So everything from seed collection and propagation and translocation right the way through to the monitoring program. And a nature refuge has also been placed over those areas to protect them in perpetuity. So that's a quick squiz at the site. This site is not on there, neither is this one, but you can see the wonderful connectivity between Apple Tree Park and other sites in Springbrook. It is essentially this area here. And this is an updated photo, so you can still see there's still some open areas to be managed. And this was part of the restoration plan, was really breaking that site up into zones, or precincts, zones, sub-zones, to really try and assist systematic work. Really essential when you have some of the weed issues that are impacting this site. So I'm just going to run through these because I also, apart from the Great Threatened Species program that's going there, I think one of the things we need to do as a group of people is really look at the variable ways we can get funding to facilitate greater restoration as well as protect our threatened species. So in the 2-for-1 requirement for the offset, 857 spiny gardenias were installed, are still alive, which is still above the 2-for-1 requirement. And in order to try and assist with some of that natural attrition, we actually popped in an extra 30% of those threatened species in day one. And with the spiny gardenia, because they have proven to be the most challenging out of the 4 species placed on site, there's probably going to be some additional planting with them down the track. So there's 23 plots created, and that's one of the before and after shots, and that's a little more recent in 2013. You can see that same plot on the right. Obviously management's ongoing, so not only weed management, but also a little bit of management around common cyssus coming in and wanting to fill those gaps. Annual monitoring is also part of this program. And you can see a couple of shots there. The onion cedar, fabulous tree, 106 plants installed over 4 plots. Again, all those areas where these plants are installed were impacted by weeds. Currently a 97% survival rate, so that's 39 plants above that 2-to-1 ratio. Again, with ongoing management, weed control and monitoring occurring. And the shiny plectranthus, a little bit of a success story in this particular project. So 80 plants were required to meet that 2-for-1 status. In 2013, the monitoring approach by the federal government said, look, we'll go to an observation status now, because more than 133 plants were surviving at that time with really clear indications that these plants were flowering, fruiting and reproducing. And so every time it rains, there's a little bit of dispersal of those plants that occur on the rock shelves and occurring downstream. And in fact, since restoration, which is probably a really good point to make, is also occurring on another creek system. So somehow they have come in, not via the same system, but have germinated post-restoration. And the Macadamia tetraphylla. I've got to acknowledge actually Hamish Corbett in this. When the plants came out of the nursery, he had enough skill and knowledge to go, that particular one and that particular one looks like it's hybridised a bit. So it was because we got a specialist macadamia grower in, so we had to cull a whole lot right at the beginning, do some more propagation translocation. Fabulously, there are 102 plants still growing, and this year some of those trees were observed to be fruiting. So all of these plants require monitoring every year to see whether they are self-sustaining. So that is reproducing viable seed. So it's going to be a few more years yet to determine when this project can come off monitoring. So you can see just a few before and afters there. So some of the additional results that have occurred so far is not only has there been some great management around these propagated and translocated species, but it's actually increased threatened species of many other plants and fauna visiting the site. The site has improved significantly in terms of its resilience. More than 280 hectares are under active restoration maintenance at the moment, and I'll show some before and afters. Some of the photographers are actually in the audience, which is wonderful. And there's been a great improvement to some of the infrastructure around the site. So, fire trails, creek crossings, the ability to access sites and continue to manage these areas. 10 prescribed burns have been implemented. And that is not only just a burn popped in, really, really good planning, great site preparation, and key to it all, good follow-up weed control. So rather than molasses grass and lantana and the like reshooting and, filling those gaps again, it's actually germinating native grasses and a whole lot of natives coming back in their place. So a bit of a win-win there. It's also been a really great demonstration site to the wider community. The QPWL, Queensland Parks and Wildlife Great Walk, goes through that site. And in the early days, not a lot of people thought you could restore this site using assisted natural regeneration. So it's been a really wonderful site, and now it is being studied widely. So it's really great to see. Some great partnerships have also occurred between governments, so particularly SEQ Water and City of Gold Coast, as well as the many, many contractors and Gold Coast staff that are working on that site. So at the moment, you can see on this particular diagram here, this is the site, all broken up into zones. The purple areas are where the nature refuge has been placed over certain parts of the site. That's where some of the threatened species have been installed. I can't tell you exactly where they are, or I'll have to kill you. In addition, we've got all these blue areas here are areas that are currently being worked. So not only is works occurring on that site, but also adjacent to that. So again, that continual improvement of resilience right across the area continues today. The hashed areas here is where all the burns have been occurring. So very steep country and very dry west-facing country as well. So a couple of before and after photos just to further demonstrate some of the great opportunities that this project's provided. So even though that lantana on the left in September 2009 looks dead, it's not. It was just a bit stressed, and in fact much lantana looks like that at the moment. These photos came from Bushland Restoration Services. And you can see that because the threatened species were placed in certain areas, really needed to keep creating buffers and then really starting to join up areas. So these are just a few little snapshots of taking areas from one condition through to another. Couple of other shots there. It's pretty clear, the changes, and now these sites, you know, that's already the aftershot 2012. These shots are, these areas are now quite full and developing rainforest in many areas, or wet sclerophyll in other areas, and on a great trajectory. Some other ones there. This patch on in this shot is actually an old banana farm and has often been a bit slow to get started, but that shot on June 2012 is now filled with vegetation, so that whole forest edge has moved right out. One of the great other advantages of doing these larger scale projects, if they're around, is on this site through a lot of large scale weed control, fuel reduction has been significantly reduced. So the threat of wildfire through these areas was a massive threat to not only the sensitive ecosystems on this site, but areas around it. So this is along Waterfall Creek, and some great before and afters. Some great shots here. I want to acknowledge Saul, who started this one, and I'm going to just quickly flick through them. You can see for yourself, and Darcy Forster also continuing the great work with his team there. This one I saw on one this morning and gets around because it is a wonderful shot of assisted regeneration. You can see the flooded gum in the back there soon gets enveloped by beautiful native vegetation. And again here with the flooded gum, you can see that's your constant point in this shot and soon gets done. So, just a couple of minutes to really acknowledge some wonderful people that have been involved in this. Darren Roach, I don't know if he's here, but has been sort of a really wonderfully consistent person throughout the 10 years and continues to facilitate some wonderful work through this. To the natural areas teams who have not only planted along the creek to assist connectivity across the site, but worked really tirelessly on a pretty hot and steep site. Also BRS, who really led the way in restoration. When we started, we needed some bloody good regenerators in there to prove what was possible, and they've been able to do that, as well as the EcoShore teams and at least 4 other companies that have been involved, Seqwater, and the many people have been involved today, and you know, it's 280 hectares so far. I'm sure the rest will get done over the next 10 years. Thanks very much.
Jen, what planting are you going to do with the threatened species? Just monoculture, or did you mix a range of species?
Yeah, great question. We just put them in the various plots. So there was a lot of work done behind the scenes to determine where they were best suited. So really trying to put them into their natural or their niche habitat. And then we just cleared really the weeds and put them in together, but various plots to try and spread that risk, really. Yeah. So together, but natives are with them now already. Yeah.
Yeah, there's more questions to ask about that, but I reckon it's ready for the next talk. We've got Paul O'Connor talking about onions.
Good afternoon, everyone. Thanks for coming. Yeah, I'm based with a community-based organisation called Environment. Based in Lismore, and we work in northeast New South Wales. Here to talk about a community conservation project targeting the emu within the Bungawalbin Catchment. Why are we interested in emus? You think they'd be common, quite ubiquitous. Well, not so in the northeast coastal strip. They've been listed as an endangered population since 2002 within the North Coast bioregion and the adjacent Port Stephens local government area. The New South Wales Scientific Committee found that the emus on the North Coast were an isolated population and had experienced severe depletion in numbers, were subject to a myriad of threats and were in imminent danger of extinction. And so just looking at the study area, basically, when it was listed, the North Coast bioregion went all the way to the border. That's changed now with the South East Queensland sort of extension. But here's the Port Stephens local government area population. There's some uncertainty about the current status of that subpopulation. I'm going to be talking about the study we've been doing within the Bungawalbin Catchment, and there are also other nearby subpopulations, the most important one being this population south of the Clarence River, which has long been the stronghold. Basically, there has been a contraction eastwards and southwards of the population south of the Clarence, and the other subpopulation between Evans Head and Iluka around Bunjalung Broadwater National Park is believed to have become locally extinct in the last 10 years. The best information we've got on emu numbers are really derived from an annual community census which the National Parks Service has been conducting up until last year. It's no longer actually been held because of diminishing numbers and difficulties around that. It obviously involves a volunteer-based survey, and so there are issues about varying effort, but the general point to take is that numbers have diminished consistently from 2010. The last observation of emus within the population north of the Clarence were around 2009. As I mentioned, wildfire and wildfire containment efforts were implicated in the demise of that subpopulation. The south of the Clarence coastal population has experienced depletion since 2012 significantly, and now is believed to be less than 40 individuals. The Bungawalbin catchment population has been a little bit more stable. We don't really have a great handle on the numbers. A lot of these are more guesstimates than actual hard and fast numbers. The low level for last year's census in the Bungawalbin was due to a low surveyor turnout due to a death of a prominent local landowner. So there are more than 7 individuals. There's a myriad of threats on the north coast. The predominant cause of mortality, or known cause of mortality over the last 15 or 20 years, has been vehicle strike. At least 73 recorded deaths in that period. We know that vertebrate pests are having a major impact through observations of feral dogs, the damage feral pigs are doing within emu habitat, and they've known to have been preying on nests down in the Clarence area, and foxes are also likely to be impacting nests and eggs and chicks. I've mentioned the difficulty surrounding small population size and the contracting population, contracting range. The scientific committee highlighted the problem of inappropriate fire regimes and long periods of increasing large areas of habitat which have had long periods of being unburnt and a consequent loss of breeding habitat. Emus nest from late autumn through winter. early spring, around the time when the land management agencies are undertaking their controlled burning management programs, so are likely to be having an effect on the population. Within the Bungawalbin catchment, habitat loss through lantana is an increasing issue, and also just barriers to movement and dispersal and associated issues of loss of access to resources are a problem. Within the Bungawalbin, it's probably more of an issue around fences limiting movement. On the coastal population, the Pacific Highway upgrade went straight through one of the core areas of the lower Clarence population. So around 2014, Envite was asked to implement a project aiming to address some of these threats. Really had three components: tried to engage and educate the community, including the local Banjalang native title holders, to increase support for emu conservation and threat control initiatives, and to implement habitat enhancement strategies to address these threats of weeds at spatially defined targets that varied according to the funding source, and also implement vertebrate pest control programs at priority sites. The seed funding was initially $30,000 from the local land services, and we got an extra 20 as part of that first stage. And on the back of that, we applied to the New South Wales Environmental Trust for a 2-year $100,000 project. Kick-started the project off with some strategic planning, consulting with the local community, particularly some of the tea tree plantations, reviewed the available data on emu records, land care group members who are quite active, and also developed a questionnaire to send out to community pickup points to try to gather information back from the community on locations of emus, their ideas about what the major threats were and whether or not they were interested to get involved in the project. In terms of the habitat enhancement work, this is sort of the result of the 2-year Environmental Trust project. When we initiated the work, we were looking at trying to concentrate works around areas where emus were known to occur and were likely to be moving, and really tried to maximise areas adjacent to tracks to increase the area available for foraging or potentially nesting. And we selected sites around a major tea tree plantation, which had an adjacent large private conservation reserve managed by the South Endeavour Trust, and also an adjacent offset property which the Roads and Maritime Services had purchased as an offset for the Pacific Highway upgrade, as well as adjacent private land owners, and also some important areas on National Parks Estate. We sought to engage the local native title community through Bogle Land Council and provide some casual employment opportunities, undertaking weed control and involvement in some of the monitoring work. Also reached out primarily through the local land care group to the broader community, enticing landowners to try to get involved through a capacity building program, weed control workshops and demonstrations. We tried to get landowners skilled up in the most cost-effective control techniques so that they could be involved in the works at every stage and undertake follow-up as well as maintenance works. We also sought to facilitate a series of capacity building workshops implemented by local land service staff at training up and accrediting landowners and land managers in vertebrate pest control techniques and methods and responsibilities associated with that. With the broader aim being to try to get to a point where there was a more strategic, cross-tenure, coordinated approach at dealing with vertebrate pest control around these priority areas. Another community initiative was the formation of a Coastal Emu Alliance group, which consisted of a number of agency representatives, community-based organisations, local landowners, wildlife carers, which periodically came together to discuss the plight of the emu in the area, share information, discuss appropriate recovery planning sort of issues, and sought to kickstart some funding applications to get money for certain activities. It was a really great collaborative sort of sharing exercise. They did successfully get some funding to look into the genetics of the coastal emu. One of the first actions we did when we commenced the local land service project was to get some signs up on the road just to warn people about the likely presence of emus and try to slow down. I'm glad that we got a lot printed because about 30% of them were removed for whatever reason, but a number of them are still there. And fortunately there haven't been that many deaths on road in the last few years. They were also seeking collaboration with the conservation agencies. Lynn Baker from Office of Environment and Heritage in New South Wales is the Saving Our Species coordinator for the emu, and she's had spectacular success with her conservation detector dog Penny, working with the bristlebird up in the border ranges. Penny has been used in the Bungawalbin area to detect emu scent and signs, another method to better understand the movement, and also hopefully to try to find a wild nest, because we have not found a wild nest, nor to the best of my knowledge has anyone in this part of the world. Haven't found one yet, but she is picking up the scent very quickly. Camera monitoring work was an important part of our project. There were really two components to that. One was around this sort of area initially managed by Reconico wildlife consultants who have been subcontracted to undertake the vertebrate pest control. And there was another one which we coordinated to better understand the emu movements. The pest control program was basically to try to identify the level and location of the feral dogs and feral pigs as a precursor to control. It consisted of generally 2 to 6 week periods of monitoring followed by a similar period of control. 1080 baiting was the main method of control. From 5 to 11 monitoring locations and around 36 bait stations were used each session. Bait and memory cards were checked either weekly or fortnightly. There was also a program of pig panel trapping and shooting to deal with feral pigs. I won't go into that too much, but there was also a soft jaw trapping and ground shooting control program for bait-shy dogs. 5 stages of control, some gaps due to inclement weather. Here's just a sort of layout of the baiting transects and the monitoring cameras in stage 3, which was autumn 2016, and some representative sort of detection rate data. Dogs and pigs were more abundant, detected more frequently than foxes and cats, and some bait uptake data showing that generally within the programs baits were more frequently taken up during the initial stages, a couple of weeks, compared to later on during the program. The overall summary from the monitoring and control for the 5 stages, I guess the level of activity was sort of proportionate to the financial resources we had for each stage. So, the first 2 stages had about as much funds available for it as the last 3. We obviously were getting, and the consultants were getting used to the equipment in the initial stage. There was a lot of false triggers, but gradually the detection sort of methodology became quite honed. The percentage of bait uptake was considered quite good, a little bit lower than what they do experience in adjacent conservation estate. Dogs and foxes were showed the highest level of bait uptake, and off targets it was mainly monitored during hot periods. The high levels of unknown uptakes were basically explainable due to periods of inclement weather where there was lots of rain, storms and wind, and fortnightly checking, and it wasn't possible to really attribute it bait take to a particular species. Generally considered successful, with the number of wild dog and fox detections decreasing rapidly over the baiting period. The conclusion was that this sort of control continues to be cost-effective, broad-scale control technique, which will continue to be successful in the future, especially if it is timed with known annual baiting programs in nearby national parks and state forests. Can't rely on it solely though, and follow-up soft jaw tracking and ground control works are necessary and were successfully applied in this project to apply additional pressure to feral dogs, and are recommended for future works. There are evidence that some individuals are bait and trap shy from our work. The M-Bite camera work was really opportunistic and trying to just get a better handle on where the emus were hanging out and moving. There's 2 emus in this shot. We were fortunate in getting detections of this pair, a male and his chick, over about 5 days and around this site, which coincided with one of the most, we used to call it the dog superhighway because it had a lot of dogs going across here. Similarly to the Ricanico work, we did encounter high levels of false triggers. Did an analysis. Basically, we had 70,000 shots, 14 out of 16 location furnished data on our target species, either emus. Okay, emus or predators of the emus, and yeah, that was 365 independent images using a 60-minute threshold for independence. Basically, our camera work did affirm the relative abundance of vertebrate pests in this area compared to the emus. It took us more than 6 months to detect an emu. We got wild dogs on the first day. I guess landowners are keen to get involved with weed control a little bit more so than getting involved with vertebrate pest control. Just like to affirm that you can't rely on one technique. You need to use a variety of techniques in this sort of integrated pest management. We didn't have the resources to tackle feral cats, but they are an increasing problem and will need to be addressed in future operations. I think just this project and others nearby have formed an excellent sort of example of community collaboration. So if there is additional funding through whatever sources, it builds on a strong platform for future investment. If we do future work, we'd look at actively removing some of those barriers to movement, fences, and probably try to get a little bit more detailed population information. Thanks very much. Lots of people to thank in so little time, but thank you.
Excellent. Thank you, Bill. Okay, we are slightly over time, but we've got a bit of spare time up our sleeves as well. So we'll carry out the session with Laura Simmons, talking about genetic monitoring of a species recovery program.
Thank you. Yeah.
Should be hiding just in there somewhere, right? Right, where is the mouse?
It's there.
I don't know. This bothers me. And we've lost our tech person.
Bear with us.
Anyone retrieve a mouse?
Bear with us, folks. Yeah, we don't seem to have a moving mouse. That's the problem.
Oh, magnetic strip. Like down here.
Huzzah for technical difficulties. Yeah, cheers. Well, it's a good thing we've got some spare time up our
Yeah,
sleeves.
exactly. I'm just going to casually run overtime.
Maybe while we're dealing with technical difficulties, since we haven't had any time for questions thus far, any questions for the speakers that are presented today? Will someone help me out here? I'm treading water.
Just a question for Pierre about have you tried to put any back into the wild yet? Have you tried to plant any back in the wild yet? Your palms?
Are you trying to plant your palms back in the wild and reproduce them? So, of those individuals growing,
I'm going to get the mouse moving.
Doesn't work unless you listen to the principles. Yeah, or here.
Your legend.
Good luck.
Continue scientist, huh?
I'm married to a scientist.
The Alliance did get some funding from the Germination Foundation to look at the genetics of the population to try to, I guess, because these are separate distinct subspecies, you might be in a better position to argue or gather funding from various sources. I'm not aware of, basically there's a lack of resources and the cost of getting potentially over, that landscape. But, there has to be probably the most cost-effective technique getting in with your coordinated better, more regularly implemented bird pest control and possibly look at how to do it getting captive through some kind of resources.
Right. Thanks for that impromptu question and answer session. And we'll just run out with Laura was talking about genetic analysis and recovery programs.
Thank you very much for having me. I'm just going to start with the caveat that I changed the title of the talk because it was originally How Did We Go? And really the only work that I did on the actual recovery program was doing bush regen with Peter Michael, who's up in the back there, Oxley Catchment group. And I wasn't actually personally responsible for anything other than the genetic analysis and some weed control. So anyway, I'd like to introduce to you Gossia gonoclada. So Gossia gonoclada is a slow-growing tree, grows to about 18 metres. It's found in dry rainforest and riverine scrubs in the greater and highly urbanised Brisbane area. So it's restricted to just a couple of catchment areas, and I'll show you a map a little bit later on. Like Pierre's palm, it was described in the 1800s and then it wasn't recorded for over 100 years. Thus, it was thought to be extinct until two lovely local botanists found one small population and then found several more in 1986. However, plant numbers at that point in the wild were less than 100. The species was listed as endangered under the Federal EPBC Act and Queensland Nature Conservation Act. As a result of this, a recovery plan was written and undertaken. So the main objectives of the recovery plan were to establish new populations and increase the numbers of individuals in existing populations, as well as preserve the genetic diversity, and that's where my interest lies. And of course, if you were at this morning's talk by Linda Broadhurst, you would know that genetic diversity underpins an organism's ability to cope with change. So really great stuff was done in this recovery plan. There was pollination and genetic studies undertaken. This informed a propagation program. As a result, 378 individuals were planted into 23 locations around 1998. If there's anyone in the room who can clarify that date, I would love to know. But as you've just noticed, the details are a little bit hazy. So this is what happens when we do some of these programs. Tags get lost. We're kind of not sure whether something was a seedling or a cutting. And most importantly, we don't really know what went where, though we do know that there was considerable effort undertaken to spread a variety of individuals from the source populations or the original plants across the recovery planting locations. So more recovery actions are actually needed for this species, and that's where our work came in. This is because there has been continued mortality of the species. So there were 73 wild individuals recorded in around 1990. Now there's 36 in the wild, and 11 of these were unconfirmed as of last year. And in fact, 2 of these, we were trying to get access to them. I had a baby and couldn't get that genetic sample. So there's also been 45% survival in the recovery plantings. However, there are ongoing threats and pressures, such as we experience in urban environments, but particularly myrtle rust. If you're not familiar with myrtle rust, it causes this lovely effect. So it's a fungal pathogen that's host-specific to plants in the Myrtaceae family. It infests new leaf growth, causes dieback, prevents flowering, seed set, and therefore we don't get regeneration of these species into the wild. However, some of the planted individuals have been observed to be more resistant. And so the aims of this study were to help the local council out with answering a few questions. Have we captured the genetic diversity from the wild in our restoration populations? And can we identify genotypes that are more resistant to rust? So here we are in Brisbane. A little bit of a zoom in. As you can see, there are, I'm going to talk to the microphone. There are 2 main clusters of Gossia gonoclada. One is in the Oxley Creek catchment here, and then the other is in the Logan area. There's the satellite imagery, and as you can see, it's highly fragmented. And these plants occur in right on alluvial terraces on creeks. So these are really vulnerable locations. Some of the wild plants that have been lost in the last couple of years have been because of flood events. We're here. I grew up here, and we mostly focused on the Logan plants. And this was because Tamara had some really great rust observations, so we could do some thorough matching of our rust observations with the genetic data. Bear with me here, I know this is overwhelming, it's genetics, and I'll make it really quick. So I extracted DNA from 102 Gossia gonoclada individuals, and that was across 12 populations. There were 23 wild individuals sampled, and these were all of the this included all of the parent trees from the original recovery action plan, and then 79 of the offspring. I used I was lucky enough to obtain DD RADseq sequence reads from colleagues at La Trobe University, and these were from closely related species Gossia, Bidwillii and punctata. I screened these to identify potential microsatellite loci, so these are neutral markers. Trialled 68 of these loci across 6 Gossia species, and as a result 24 were used in our Gossia gonoclada research. 24 were scored. Of those, 20 were polymorphic and therefore informative to our study. I looked at basic genetic diversity measures to examine restoration success, and then multi-locus genotype were assigned to each individual's. A parentage analysis was conducted in service. We split our data set into family groups and matched these with the rust observations. I'm not going to go into the rust observation methodology because it was very rigorous and I don't have time to explain it. Plus, it is actually Tamara's work, so she'll be happy to talk to you about that if you'd like to know. So overall, there was moderate genetic variation found across the species as a whole, and there was little evidence of genetic clustering or isolation. This actually fit really well with the previous results in the 1990s by Shapcott and Playford, and basically is an indicator that there was historic gene flow between all of these catchments and all of these plants. There was probably a lot more plants in the wild until we had a very rapid loss of individuals and populations of the plant as Brisbane became more urbanised. So the bar charts, oh, it's a bit faint. You'll get the gist though. The bar charts on the right there are just 3 standard genetic diversity measures. The bar on the far right-hand side is the largest population of Gossia gonoclada. Now I've added the recovery populations in green. So overall, we did find that the genetic diversity of the species has been captured with similar genetic diversity measures across all of the restored populations. So as you can see, it's approaching the highest or the most diverse population. And this is really a great success story, but maybe also points to the fact that if we had 50% loss of individuals, potentially the most genetically diverse ones survived. Getting into some of the genotyping, overall I found there were 18 private alleles. Don't worry too much about that. All that means is that these were alleles or markers that are restricted to one population. So 5 of these were shared across multiple individuals, whereas the remaining 13 were not shared, meaning they were only found in one individual. And this is really useful if you're trying to do genotyping and figuring out who's the daughter of who, because these markers are very distinct. So one of the reasons that we knew that some of these markers were real and not just a fragment of our analyses is that we did know that there were some plants that were cuttings. These markers actually occurred in both the parent and the cuttings, so we had really good kind of data quality control there. In the end, I was able to assign 65 unique breeding pedigrees, and the identity of 66 of the seedlings were resolved with 95% confidence. So this kind of genotyping allows you to come up with things like this. So the plant on the left is a cutting of the plant on the right. Also helped us identify that these plants here are seedlings and the result of a self-pollination event, whereas this seedling is the result of cross-pollination. This is very interesting if you're into this kind of stuff. And here's one of the individuals where we couldn't quite ascertain the exact parentage, but we were able to narrow it down. So of course the burning question, I'm sure, which genotypes had rust resistance. So the 65 individuals from 9 genetic groups were matched with 3 years of myrtle rust resistance observations. Genetic groups with significantly lower rust scores were actually related to 5 of the original trees. And as I mentioned before, interestingly enough, these trees had the highest levels of heterozygous loci and possessed some of the unique alleles. So this is again going back to that concept of genetic diversity is really underpinning our ability, potentially underpinning our ability to cope with environmental change. So the conservation implications and the summary of, I guess what we found is that we did find, genetic diversity was represented in the recovery populations. We were able to identify pedigrees and family groups and match these with myrtle rust resistance information. From these results, we can provide council with some really great recommendations. So in the future, when you undertake the next round of recovery plantings, to use a combination of wild and offspring individuals to maintain full genetic representation of the species. Unfortunately, some of the wild species, so the original plants, we have lost some of the largest individuals, again, mostly related to flood events. Fortunately, we've captured this genetic data with the offspring individuals, so it's really important to kind of use the full range of individuals out there for future programs. And it also gives the ability to select rust-resistant groups. So ideally, it would be great if we could add some new plants to current recovery as well as the wild populations. This would create a mixed age structure and increase generational security. Another kind of sad thing about Gossia gonoclada is that there has been no recorded seedling or seed germination and new recruits into any populations since they've been monitored in since the 1980s. Unless you know any, Leanne? Maybe?
Yeah, maybe.
Yeah.
Maybe.
And also that propagation by cuttings may be ideal, while this sometimes is not the preferred method in restoration. In this case, it might be ideal and really the best way to go about it because it gives the ability to select known genetic material as well as overcome the limitations of low fruit set for the species. And there's a whole suite of people to thank. And yes, that's me. Thank you.
So that brings us to the end. We can spend a couple of minutes posting questions here, or alternatively there will be coffee outside. I'm sure you can jump over coffee as well. So, yeah, one question for Patrick?
Yeah. Thanks, Laura. Are you able to say at this stage, are there any shipping light on issues we have about some of the other endangered plants in the basin? I'm thinking mostly both nervous and also rhododendron, which are obviously critically endangered at this point. About what we know about the genes, do you think there's some research coming up about the similarity of genes that remove rust resistance?
Yeah. Certainly. Okay, so there is someone here who has been studying, but he's not in the room at the moment, but he's presenting in the next session. Jarrah Wills, he has just spent a year basically doing a lot of work on all of the different myrtaceae that are affected by myrtle rust. Certainly, I also know that there has been some work done on the genes that make plants more vulnerable to myrtle rust. And one of the reasons that I trialled markers across a variety of different Gossias was because I was quite interested in the fact that if we were successful in using the markers that we used, we could transfer them to other species. So yeah, definitely I think there's a place, and hopefully there will be some work done on some of the genetic I guess, background of those other endangered species. Yeah, particularly to inform future translocations to like protect their future. Oh, that's a very good question. I'll have to look at my data set for that one. Yeah. I actually found out that a lot of the seeds were actually from the pollination program, so I'd like to do a bit more tracking through old records to see what I can match up and, yeah, look at that. Thank you.